Good morning, everyone.
Good morning,
Yeah,
sir.
Roshni, you may continue, ma.
Okay, sir. Yes, sir.
I am leaving from the meeting. You will end.
Okay. Right. Hello. Yes. Good morning, everyone. I'm audible, right?
Yes, ma'am.
Okay. My voice is clear. Yesterday it was very low. No, now it is clear.
Yes, ma'am, it's clear.
Okay. Yeah. So we all know Brassica oleracea variety botrytis means the cauliflower. So which is very widely grown in all the continents of the world. As I have told you that there is a lot of diversity because it was a leafy. It was from Brassica oleracea variety sylvestris, which is the leafy kale from which all these cole crops originated. So it was a leafy type that evolving into a leafy head is different. That evolving into a flower means this is an inflorescence that we eat is another different thing. There's a lot of what call mutation, introgression, evolution, and human selection that played very much role in this complete process. Nextly, probably the kale was the first coal crop to be selected and adapted by man. So among all the coal crops, kale was the first crop that was selected and then adapted by humans. And the use of kale as human food consumption was even mentioned by Theophrastus in 350 BC. And Greeks in 600 BC. So it is a very old crop. From the leafy kale-like plant, other coal crops like kale, collard, broccoli, cauliflower, and cabbage have descended. And the nuclear RFLP studies have told that all these have monophyletic origin, means all have come only from one single ancestor. It has given like that. So it has also been shown that the 1,000-headed kale has come from the Brassica oleracea. The marrow stem kale or the stem kale came from Rupestris incana. And the branching stem kale has come from Brassica critica. And the wild leafy kale has developed from variety costata. And the ancestral broccoli, which is nothing but means the cultivated broccoli is there. No, the ancestor, just the immediate ancestor for that cultivated broccoli is Brassica critica. So this Brassica critica is also a wild annual. So what we know, all the coal crops are biennials. We know that they are biennials, but this ancestral broccoli means the just predecessor of broccoli, which was annual in nature with cymose white flowers. It is cymose inflorescence even now. Okay, so this is a primitive form of broccoli which is cultivated and evolved several thousands years ago. So from that to evolve as the broccoli that we have today, it took some thousands of the years. The ancestral form of broccoli, when it moved to southern China and then gave rise to the leafy kale types of the Chinese kale. So we also know that Chinese kale is there, no? So this Chinese kale has come from this broccoli in China. Chinese kale I am talking. So the ancestral sylvestris is one of the leafy type of kale only. But what happened? The Chinese kale, that is Chinese kale, Chinese cabbage are different. The scientific names are different. So all these have originated from this broccoli. The which broccoli? The primitive broccoli, that is Brassica critica, not this Italica. Okay. So natural crossing between ancestral broccoli and the biennial species, wild types, have resulted to the development of this Brassica oleracea species, which include the broccoli, including the sprouting broccoli and various other forms of cauliflower also. A natural cross between the ancestral broccoli, that is Brassica critica, into some other wild biennial forms has given rise to the Italica broccoli and also this Botrytis cauliflower. That is why now they have got the shape and the structure as the cauliflower or broccoli, having the same form and structure with a biennial nature, because it was outcrossed with some of the wild biennial types. Okay. And sprouting broccoli was probably used in early Christian era, but it was not documented till 1660s. So up to 1660s, it was there was no record of broccoli, but it was said to be used. It was before also. Okay. And cauliflower, comparatively, it originated very later. First broccoli originated, after that cauliflower came about 500 years ago, just 500 years ago. Even in India also, cauliflower came very lately. Probably by the introgression with broccoli gene pool, this cauliflower has also came up. And Syria is considered to be the place of origin of cauliflower. In general, cole crops are originated from Mediterranean region. I don't say you means, "Arey, ma'am, you are telling cauliflower is from Syria." It's not like that. Here, when we discuss the history, we see that cauliflower has come from broccoli. Broccoli has come from kale, the wild kale. But the Chinese kale is also come one of the selection from this broccoli. But all the ancestral broccoli which we are talking, the scientific name is Brassica critica. It is not Brassica oleracea variety italica. So you have to understand the complete tree from which species which has come. Okay. And nextly, so in this process of evolution, the exact cauliflower that we eat today came from Syria. Okay. And it was probably introduced in Italy around 1490, means in the 15th century only. That where is where is 350 BC and where is this 1490, means 15th century you have come. So how many thousands of years this has been evolving, you have to understand. Okay. And Italy is a center of diversity for cauliflower and also for broccoli, because now several land races are present in that particular place. And at the beginning of the 17th century, cauliflower reached Germany, France, England. And as all the flower buds open in the sprouting broccoli, compared to the prefloral fleshy apical meristem of the cauliflower, it is thought by many that cauliflower must have preceded the broccoli, but it is not like that. What we think, because if I don't know if you are understanding or not. See, broccoli has this inflorescence with open flowers, but this cauliflower has stem only, the fleshy apical meristem stem cauliflower. So what we think, from cauliflower broccoli has come, but from broccoli only cauliflower has come. That is what you have to understand. And nextly, probably this Romans and others introduced this ancient kale, leafy cabbage, broccoli, cauliflower in England and other Mediterranean region. From Mediterranean region, these Romans have taken these kale, cabbage, broccoli, cauliflower, and they have moved it to England, France, other parts. Okay. And cauliflower in India, how did it originate? While what happened, while it was taken from one place to another by this predator, means these people, when it was taken, what happened? This cauliflower or whatever crop it is taken, they started adapted to the conditions. They became winter hardy because it was taken from one mild climate to very cold climates. There they became winter hardy, and from there, because it was crossed to a biennial wild one, it became biennial in nature, and their inflorescence was racemose kind as the ancestral kale, whereas only broccoli was having a cymose inflorescence, others were having racemose inflorescence. So even a lot of variation, lot of adaptation to the conditions started in the coal crop. And then what happened? Cauliflower was introduced to India from England. So I told you, as Romans took those to England and France and other, from England it came to India. In this point is very important. Cauliflower was introduced to India from England in 1822 by Dr. Jemson. This complete line is important. Cauliflower was introduced into India from England in 1822 by Dr. Jemson. So he was a botanist in the Kew Garden, okay, from Kew. So and in charge of the Company Bhag, Saharanpur, United Province, now called the Uttar Pradesh. So he was in charge of the Company Bhag, the garden there. So the Royal Agri Horticultural Society, Calcutta, also introduced cauliflower and other English vegetables in 1824 from South Africa. So from the Royal Agri Horti Society, also they introduced, but it was introduced in 1824. Before only this Mr. Dr. Jemson in 1822 itself, he brought it from England. Okay. And kohlrabi, the knoll kohl that is there, no, the kohlrabi and the Brussels sprout were introduced later. First cauliflower came, and afterwards this what you call this kohlrabi and Brussels sprout have come to India. So broccoli is a recent introduction, which again came very lately. In 1990s, it was brought from USA and Japan for commercial cultivation in India. That is very recently it has been brought. But cauliflower also is a recent introduction, only 1824. Because I remember very nicely that first time means it has means just in 1824 if it is coming to India, it doesn't mean by 1825 it will be spread all over India. No, it will not so fastly spread all over country. It was my grandmother who in our family generation, who my very own grandmother was the first to you means she saw cauliflower for the first time when she was some 24 years or 25 years it seems, and she did not know how to cook it, because like that was the first time she was seeing the vegetable. But now everybody knows cauliflower, so it has got domesticated. My grandmother means just immediate grandmother only. So it is very recent. Cauliflower, broccoli and all are so the Cornish cauliflower of England was the first to be introduced. So in cauliflower also the Cornish cauliflower was first, and later it got adapted to high temperatures and humid conditions of India, and then the tropical Indian cauliflowers were developed from those Cornish and European type of cauliflowers. And now that we have this cauliflowers that can be grown in even 12 to 16 degrees, 16 to 21 degrees, 21 to 25 degrees, and 25 to 30 degrees also. We have this wide range that has been developed. So in India also, first which cauliflower came? Cornish came, and after that, after the adaptation to high temperature and humid conditions, we have got the cauliflowers that we are consuming today in India, is in South or North India, whatever it is. So natural crossing between different types are followed by farmers. In the process of selection, what happens? Natural crossing occurs because it's a highly cross-pollinated crop, no? So natural crossing occurs, a lot of adaptation to the tropical conditions will occur. And because whatever cauliflower was brought to India were European, means they were temperate type of cauliflowers. So these several Indian cauliflowers, which are now they vary in the period of maturity, that I have told you now only, no, that 12 to 16, 16 to 12, like they're varying in maturity periods has been developed, and based on the season, See, it is, we call it 12 to 16, 16 to 20, 20 to 25, 25 to 30. But the thing is, based on the season, they have different names called, means Kanvari, Katki, Aghani, Pusi, means they are the seasons, means early, mid late, late. So this names Kanvari, Katki, Aghani, Pusi means the early, mid, late, mid late kind of things. Okay. So, and also on the other hand, with the maturity period varying, it also has some tolerance to temperature conditions. Okay. Besides being heat tolerant, Indian cauliflowers have also developed some of the resistance also in this process of evolution for downy mildew disease, which is Pernospora parasitica, black rot, Xanthomonas campestris, and curd and inflorescence blight, Alternaria brassicola. In the early maturing Indian cauliflowers have very stable self-incompatibility, which is not thermosensitive like other temperate European cauliflower types. Means our Indian cultivars have a very high amount of self-incompatibility compared to the European types. Okay, Indian cauliflowers or our native cauliflowers have high self-incompatibility. Nextly, what happens, coming to the biosystematics, that is the chromosome number and all. So the family is Brassicae, the n equals to 6 to n equals to 75. Such means in this process of evolution, you have n equal to 6 to n equal to 75. And there are more than 220 species and 52 genera in it. And yesterday also I have told you the basic chromosome number being 18, and Brassica oleracea belongs to C genome where n equal to 9. Okay. Next is genetic diversity. So coming to the genetic diversity, as it has it is a main origin in Mediterranean region, but whereas cauliflower came from Syria, and different means, whereas in China this Chinese kale evolved. So based on different regions, there are many, all the Western countries, every country has its own adaptation of cauliflower there. So there is a different story everywhere. So the coming to genetic diversity, it is just it has evolved there, it has many land races there, all kind of things. That's not so important because only place names will come, because you can see the in the 16th century, this Erfurt, Alpha, Snowball kind of things came in Germany, and the European biennials, very important varieties like Old English, Roscoff, Angers, they all came from Europe, and like that only. All the varieties are there, not so important, but there are some famous varieties, this Erfurt and all. Okay, coming to the genetics that I told you, that is very important in every time I tell you. So some of the traits that are governed by polygenes include polygenes like the leaf characters, means if leaf is glossy, how the leaf arrangement is there, what is the stalk length, curd color, flower color, all these are included by polygenes. Okay, polygenes. And what are monogenic characters? So perhaps the loosiness, rissiness are highly heritable natures, and they are governed by a single gene. Rissiness and all are governed by a single gene, monogenic. And some curd color also, orange color is governed by single gene, whereas green color is governed by two genes. And coming to the leaf arrangement, means around the curd, how the leaves are arranged, means two genes are involved in this arrangement of the leaves, how the arranged leaves should be arranged. And coming to the flower color, which is white, yellow, cream, so these three variants of flower colors are present in that two independent genes are involved with dominance and epistatic effect. And coming to the male sterility, the transgenic Barnes, bar star. If you don't know, you take BD Singh book, you will understand what is Barnes, bar star. If not, you can just Google you can Google and read, or you can just search in YouTube. You will find Barnes, bar star videos, animated videos also. What are this Barnes, bar star genes, and what is the transgenic, means how that, what is the action of it, you will understand if you don't know that. It is a BSc question actually, so it should be covered in your BSc. If not, please see in a animated YouTube video, you will understand better what is Barnes bar star genes. So these transgenic cauliflowers through that system have also been developed. And next is there means Barnes bar type of male sterility. It's a kind of male sterility only, induction only. You just see, you will understand. And generally, genetic male sterility is controlled by a single recessive gene, which has been developed. Reported both in broccoli and cauliflower. So a dominant gene of male sterility occurs in broccoli, but in the case of cauliflower, a duplicate dominant gene with cumulative effect, means additive effect, is found in case of cauliflower for the sterility. Okay, directly a single dominant male sterile gene is present, dominant male sterile gene is present in broccoli, but whereas for cauliflower, you have duplicate dominant genes, that too with cumulative effect, additive effect, means directly one single gene is not involved, some many genes are involved, but they are dominant only. So that is there for cauliflower. But in general, GMS with the recessive genes is being reported actually. Okay, however, male sterility is not commercially used in production of hybrid seeds because the maintenance and multiplication is uneconomical in case of coal crops. And even micropropagation for male sterility, multiplication, means maintenance of male sterility and all also are used, means micropropagation is also done for the hybrid seed production. Next, for male sterility to maintain and all, you all go for micropropagation also. Coming to the self-incompatibility, as I have already mentioned, that the self-incompatibility in case of Indian cauliflowers is strong than the European cauliflowers. I hope everybody is listening. Nobody is sleeping, no?
Yes, ma'am.
Yeah, okay.
Yes, ma'am.
So Indian cauliflowers have more self-incompatibility than the Bhavesh is not joining, huh? Because it is very early for him, maybe. So, what am I? Okay. And the self-incompatibility in Indian cauliflowers is very high compared to the European cauliflowers. And most of the Indian cauliflowers, we know that they are heat-tolerant cauliflowers. That means you can call them, instead of cauliflower, you can call them Indian cauliflowers, because this cauliflower is completely different from what is in Europe or the Mediterranean region, because they are completely maybe different because of the traits that they have, characters that they have. Okay, so these Indian cauliflowers spore also, means all have this sporophytic self-incompatibility, which is governed by multiple S alleles. So with the dominance relationship, okay, this is the highly dominant relationship, means they are dominant in nature. And there also we have S1, S2, S3, S4. You might have seen a table where S1 is greater than S2, S2 is greater than S3. That table also you might have seen. It is nothing but it is the kind, see, there are many S alleles, multiple S alleles involved in self-incompatibility. So alternate form of a gene is nothing but allele, no, means that the particular gene is, allele means what? For my development of my eyes, only one gene is involved, but that gene may have different forms within the gene. What are the different forms? They may be yellow, green, orange. So if some G allele is involved, gene is same, but G allele is involved, then my eye will be green. O allele is involved, then the gene is same, but my allele is O, so my eye will be orange. So there are different alleles of the gene. So the alleles may be having various various traits. It may influence various traits. Similarly, this self-incompatibility has many alleles, but all are S alleles only. So what we gave S1, S2, S3, S4, like that, we gave numbers to those S alleles. So if you sometimes what happens, S1, so what happens, S1 is the weakest self-incompatibility and S4 is the highest self-incompatibility. So in case in that particular plant S1 is greater than S2, means it is comparatively less self-incompatible. Because S1 has lowest self-incompatibility, that's S1 allele, whereas S4 means as you increase the number, it has high self-incompatibility compared. So that's why what happens, you write S1 greater than S2 greater than S3 greater than S4, you write. Why you write like that? Because S1 has less self-incompatibility. If because it is the same gene, no, if S1 is there in that plant, it is less self-incompatible. If a plant has S3, then it is more self-incompatible than this first plant. That is the reason why you write S1 greater than S2 greater than S3 greater than S4. What is this S? You may think this is the because this sporophytic self-incompatibility is governed by multi-allelic, means it is the same gene, but it has multiple alleles, and all the alleles are S only. They are not, see, I was telling green I means G, orange I means O, but for this all are S alleles only. So lot of S are there. So you will get confused, no? So the numbers have been given S1, S2, S3, S4. And in that S1, S2, S3, S4, what happened? S1 has the weakest self-incompatibility, and as the number increases, self-incompatibility means the strength of self-incompatibility increases. That's why we write S1 greater than S2 greater than S3 greater than S4. I hope you understand. If you don't understand, I can't help you. So the commonly grown snowball variety is self-incompatible. In self-incompatible plants, the incompatibility is effective 2 to 3 days before anthesis. And hence what happens, if you do bud pollination, it becomes a self-pollinated only. You understood what I told you? What the self-incompatible nature is there, no? That will work only just before, means if just before 2 to 3 days of flower opening only. From 2 to 3 days before flower opening only, this self-incompatibility will come into play. So if before 2 to 3 days of flower opening, if you pollinate, then it will self-pollination. Okay, that is immature bud. If you pollinate an immature bud in case of cole crops, it will become what you call, it becomes self-pollinated. It will be compatible only. But from 2 to 3 days before pollination onwards, the self-incompatibility comes into play. And then you, if you pollinate also, it will not take, it will not accept. And after flower opening also, it will not accept. If it is immature bud means, if you pollinate, it will accept that, it will become compatible. So incompatible plants show their incompatibility from 2 to 3 days before anthesis only. Hence, if you want to do bud pollination, if you do bud pollination, they are self-pollinated only. They can be compatible. Okay, the S alleles that I told you, that there are multiple S alleles, these are highly environment and temperature sensitive. But in case of Indian cauliflowers, they are less temperature sensitive. Means the self-incompatibility in case of European cauliflowers, if you give high temperatures, the self-incompatibility will break. But in Indian cauliflowers, if you give high temperature also, the self-incompatibility will not break so easily. Okay, that is one case. But in general, what happens, this self-incompatibility in general is also influenced by the environment, especially temperature. So what happens to break the self-incompatibility in Indian cauliflowers? You need to give more temperature. Sometimes it may work, sometimes it may not work also, because it has a stronger self-incompatibility. To overcome this self-incompatibility, if you want, because if you have some certain favorable trait in that particular cauliflower plant or cabbage plant, or so you cannot self it because it is self-incompatible, but you want to maintain that particular plant. How you will maintain? So you have to break that self-incompatibility. So you are going and doing bud pollination, because only 2 to 3 days before only this self incompatibility will start. So what you will do, the immature bud pollination, and still it is not happening. Means there are different methods, like if you have rub salt on this stigma surface, or if you apply excess CO2, and these are some of the methods, giving extra temperature, and these kind of things, double pollination, all these methods will help to overcome the self incompatibility. Actually, I don't know if you people have read or not in BSc and all, it's an important question, how to overcome self incompatibility. Okay. Next, the CMS system. So we know that in cabbage and all, we read Augura cytoplasm, Anand cytoplasm, so like that only. The CMS system, in case of cauliflower, the Augura cytoplasm from radish is most important sterile cytoplasm that is used for cauliflower, broccoli, and cabbage also. So the hybrid that was utilized to transfer the CMS to cauliflower from Augura radish. Okay, the CMS has been transferred from Augura radish into cauliflower. However, it had problems of temperature sensitivity. I told you in case of cabbage also, there is like chlorosis. If you cross this cabbage male with the radish female, you will have this plants which are developed having severe chlorosis. Here in cauliflower also, you observe that they are highly sensitive to temperature, and they have a high amount of chlorosis when you cross it with this Augura cytoplasmic radish. Okay, and now to overcome this problem of chlorosis, they did a protoplast fusion method also. And then this Anand cytoplasm is there, no? And it's to overcome this chlorosis, means the chlorosis is not a favorable character. All the plants are yellow means what is the use? Nobody will buy your cauliflowers, no? So that's why what happened, they went for protoplast method of fusion of two. Protoplast fusion means what? The protoplasts of both cauliflower and radish have been fused, and then a hybrid has been developed. Now after this protoplasmic fusion, the cytoplasm of radish has come into cauliflower. So now what you will do, this cauliflower has been used. Such cauliflowers are now used for the hybrid seed production of cauliflowers. Okay, and to transfer this sterile Anand cytoplasm from Brassica rapa, so what they have done, they have used this wild species Brassica tourniforte they have crossed this Brassica tourniforte to broccoli and cauliflower, and a new source for producing hybrids has been developed from Anand. Anand cytoplasm, sterile cytoplasm, has also been slowly drawn into this cauliflower. A recessive male sterile gene. Overall, this male sterility and all are governed by recessive male sterile gene in cauliflower. Whereas in GMS, I told you, partially dominant in cauliflower, dominant in case of broccoli have been found. But for CMS, recessive MS genes have been found. And coming to the resistance to diseases, so there are many important diseases like clubroot, that is Plasmodioptera brassicae, Alternaria leaf spot, Alternaria brassicae, the curd and inflorescence blight, Alternaria brassicola, and downy mildew, Pernospora parasitica, white blight and water spot, that is Sclerotonia sclerotorium, black rot, Xanthomonas campestris, soft rot, Erevenia carotovera, and the turnip mosaic virus, root knot nematode. So including this resistance to this clubroot in Brassica oleracea, this broccoli, cabbage, so all in any cole crops, this all diseases, and especially this clubroot disease, they are governed by two major recessive genes and two major dominant genes. So two major recessive and two major dominant genes are involved for the disease of clubroot. Okay, and resistance, they in case of diseases also, what happens, like in virus you say strains, for this bacterial diseases, you have this race 1, race 2, means in virus you call, no, this is strain, Indian coronavirus strain, it has mutated, so you call them strains. When they mutate, they are the same virus but they have different forms, they're based on the location, condition and environment influence, no. They similarly for bacteria and all, fungi and all, you have this race 1, race 2, race 3. So resistance to race 6 and 7 is controlled by the single dominant gene, and resistant to race 1 and 3 are governed by interacting genes. So some genes interact together and provide the resistance. So this is for club root. There two dominant, two recessive genes. In that race 6 and race 7 are governed by single dominant gene, whereas race 1 and race 3 are governed by some interacting genes. Okay, and nextly what happens, resistance to this Alternaria brassicola, that is nothing but their curd and inflorescence blight in case of the Indian cauliflower, was found to be polygenic in nature, and also some dominant and additive gene effect also has been observed. The resistance to downy mildew, the race 1 and race 2 are controlled by single dominant genes, and whereas Indian cauliflowers selected lines like MGS23, some lines are there, they are highly resistant to downy mildew. Some lines like MGS series, in that series 1612, 12C, all these lines are reported to be highly resistant to this downy mildew disease. Okay, and similarly, resistance to the white blight or the watery soft rot in cauliflower is controlled by major recessive genes along with modifiers, and resistance to the black rot was found to be dominant. Resistance to black rot is dominant over the susceptibility. So black rot is a dominant gene, so you can easily incorporate the resistance to black rot. And nextly, the dominance, this is all not important, many diseases you will get confused. Okay, coming to resistance to insect and pests, so a glossy leaf surface of the cauliflower will help us for resistance against lepidopteran pests. Okay, glossy leaf. Okay. Next, however, this glossy leaf character reported in Indian cauliflower did not show any resistance to the insect pests. So glossy nature, generally we know that it provides resistance to pests and diseases because it will, the surface will reflect. But whereas what happened in those surface, the glossiness that is present in Indian cauliflowers is not able to resist any insects and pests. Okay, and nextly coming to the anther culture, so double haploid lines have been developed using this anther culture in case of cauliflower, and this double haploid lines means what? The haploid has been doubled, no? So genetically they will be homozygous in condition. So this they are commercially used for hybrid seed productions in production in India, it seems. And nextly, coming to the breeding objectives, now I want somebody to give me good breeding objectives. Who will give me breeding objectives, good breeding objectives? I told you have to use some chemical compounds. So the curd flavor, which is an important character, so which is the volatile chemical compounds like nitriles, isothiocyanides, and this enzyme that myrinose, which hydrolyzes this glucosianides, impart flavor to the curd. So those compounds which impart flavor should be increased. And during the domestication, what happened? The non-bitter taste due to low glucosinolate content has evolved. So you have to see that the glucosinolates, the harmful compound should be less. The taste inducing compounds should be more. And heat tolerant cultivars generally are little bitter in taste. So that heat tolerance with the reduced bitterness should be, we should search for that kind of things. And apart from that, when quality comes, not the yield just means yield more, and biotic and abiotic stresses, if you leave, what happens? The curd diameter should be more, the curd color, it should not be yellow, riciness should not be there, the flowers, the head should not become opened, loose head should, sorry, curd, the loose curd should not be there. So the diameter and all, the shape and all is very important, compactness is important, weight is important, shape is important, color, maturity. So the early, late, mid maturity, every stage of maturity has its own curd characters. So that curd characters of every stage has to be improved. Means even though it is early, it should not be loose, like that. So the nextly, erect and semi-erect kind of cauliflowers are observed in India. Okay, so the semi-erect type is more efficient than the erect type, it seems. So while you grow, what happens? The semi-erect type is better performing than the erect ones. So you should use the semi-erect ones. And a self-blanching kind of thing is very, very important. Self-blanching kind of cauliflower you need. So this is all important points. And nextly, what happens? Resistance to disease and pest. Instead of that, in cauliflower, if you write this resistance or tolerance to heat, to frost, high soil moisture, high humidity, Indian conditions you write, Indian conditions. So then it will, it is like, yes, there are some good breeding objectives that I'm expected, you are expected to write in exam. And by what are the common, in case of coal crops, what are the common pollinator? Honeybee is the common pollinator. Okay. And coming to the breeding method, as I have already mentioned, in case of cabbage, there are many breeding methods, like because it's a cross-pollinated crop, and with many systems like CMS, GMS, and the self-incompatibility. So you can go for many kinds of this breeding methods. Almost all the breeding methods can come into play. And even if you want to self it, you can go for the bud pollination. You can break the self-compatibility, and you can do whatever you want to do with it. And in case of this cauliflowers, even in case of cabbage also, synthetic varieties are very, very important. And in case of cauliflower, you have more synthetic varieties than the cabbage. Okay, so this synthetic varieties involved almost 6 parental lines are involved, and those 6 parental lines are left to cross in between themselves. So all the 6 will be having whatever favorable traits you want. They will be having all the favorable traits, and they will be self-incompatible. So they have to only cross within each other, those 6 only. So 1 will cross with 2, 2 will cross with 3, 3 will cross with 6, like that. So ultimately, after that, you will harvest the seed and you will sow that, and then you will see which is performing nicely, and then you will release it. Okay, and some of the so as the temperature range, in which temperature range, what are the varieties, and what is the maturity stage, that is early, mid late, late, all this you have to remember. You have in your glosses or anywhere also you can take, and you can remember the period, the temperature, the varieties that come under that particular section. Okay, after that, there are many local varieties, not in India. Indian local varieties are also there, because as I told you, Kanwari, Katki, Aghani, Pusi, Maghi. So in exam, Maghi belongs to which section, late, mid, late, like that they will ask. Okay, so like that, you have to know that also. They will be given in brackets in case of, I don't remember in glosses they have given or not, but they are the name of the seasons. Whereas nextly, coming to the area and production, it will change, you can learn afterwards. So the coming to the common general production technology, in September, whatever we start growing, the maturity group which we can harvest in September itself, where the temperature is 20 to 27 degrees or 30 degrees. And in October maturity, if it comes to 20 to 25 degrees, in November maturity comes 15 to 20 degrees, and if in December the maturity comes, then it is 12 to 15 degrees. Whereas the late snowball types, which are typical European cauliflowers, they require a temperature of 10 to 15 degrees. Okay, and nextly, it is a transplanted crop, we all know, and how to make a bed 15 centimeter high, 60 centimeter wide, that all also a basic information that you can read from the basic horticulture book. Okay, Jitendra Singh book. So in general, the seed rate will be 500 to 750 grams per hectare, and the spacing will be 60 into 30 centimeters for the general planting. And for early varieties, also you can go for less spacing only, because they will mature early, so their size and weight and all will be very less. They will not grow vigorously, early varieties, so you can grow for 60 to 30 centimeters. Whereas as the season improves, means for the mid season, late season, you can go in for 60 into 45 or 60 into 60 centimeters, because they will be more vigorously growing than the early ones. Okay, and generally for rainy season and all, ridge and furrow method is preferred, not than flat bed. This is common sense. Why in ridge and furrow only in rainy season? Because the rain can go from the furrow, it will not damage the crop. It is flat bed, means completely crop will be damaged. Okay, and nextly, coming to the intercultural operations, blanching is a very important intercultural operation, that is covering of the curd with the upper leaves or some paper or polythene, just to make sure that the curd is not exposed to high temperature and it does not turn yellow. We go for blanching operation, and this blanching is a very important operation. And apart from that, earthing up so as to support the growing curd, and also weeding and all other operations. So average yield is 10 to 12 hectares, and late and mid-late varieties will give you up to 20 to 30 tons per hectare of yield. And because early varieties will have smaller size curd, whereas the whereas this late ones will have larger curds. And in general, in open conditions, you can store for 3 to 4 days, and 0 degrees Celsius you can store 30 days only. Okay, whereas you have cabbage, you can store for more number of days, but in case of cauliflower, you can store it only for 30 days or 1 month, even in 0 degrees Celsius. So the varieties in early section, like early Kanwari, Pusa, early synthetic, Pant Gobi 3, the October maturity groups, you can go for the Pusa Katki, Pusa Deepali, RCJOB 1, the November maturity groups, like Pant Gobi 4, Punjab Giant 26, Punjab Giant 35, Hisar 1, Pusa Sharad, Pusa Hybrid 2, like that. And the December maturity group, Pusa Shubhra, Pusa Synthetic, Pusa Himajyoti, Pant Shubhra. And the late maturity group is Pusa Snowball K1, Pusa Snowball K1. Okay. So with this, rest you have to know. And tomorrow we will once cover all the 3: broccoli, nolcol, and rabi, Brussels sprout. All the 3 we will complete tomorrow itself, because they are small ones. Okay, some basic information of how and from where what has originated. So coal crops have originated from coal. What coal means colis means stem, like that. Instead of just directly by reading your Gloucester's instant or essence, you some basic general knowledge of the crop, the origin, and when it came to India, by whom it came to India, and all. In case of tomato also, Royal Agri Horti Society has brought tomato also. Similarly, Royal Agri Horti Society Calcutta has only brought this coal crops or cauliflower to India, but it was first by Dr. Jemison. So all this general things will help you to understand the crop better, and will improve the overall knowledge of the crop also. Okay, any doubts you have, or I can close this class? Any doubts? You have to read, just listening will not help you. You have to read by heart things, then only you can answer in exam. Any doubt anybody has?
No, ma'am.
Okay, then I will close the section. Yes. Okay, then all the best. Read well. Bye.
Thank you, ma'am.
Thank you, ma'am.
Thank you.